Good morning, and thank you for joining us. It's my pleasure to introduce today's speaker, Professor Colin Campbell from the Department of Pharmacology. Colin received his PhD at Boston University and his postdoctoral training at University of Illinois at Chicago and also Albert Einstein College of Medicine in New York. And he joined University of Minnesota in 1992. Building a successful research program investigating mechanisms of DNA repair in mitochondria and also in nuclear DNA. He will be talking about his project today, but I want to also mention that Colin has been very involved in university governance and student training here at the university. So he assumed leadership roles in bringing in funding and making it possible for minority students to come and do research in the Department of Pharmacology and obtain funding for this program. It's a life sciences, right? A minority program. So that's a great thing. And he also is chair of University Faculty Consultative Committee at the University Senate, and it's his second term. And he says that's what you do if you, committed something in your previous life, because it consumes his life, and we want him more in the lab. But I think it's a very important leadership role that he's doing, providing the link between faculty and university administration. So today, Colin will talk about a project that actually our two labs have been collaborating on for the last 15 years. It's scary. He was the first person who actually trained me about cell culture and a lot of biological techniques we didn't know how to do. And we've had a very productive collaboration that is now supported by an R01 from NIH. So welcome, Colin, and great to have you here. And he will talk about repair of DNA protein crosslinks.
Wow, it's a rare treat to have a really respected and long-time collaborator introduce you. So, Natalia, thanks very much. I was going to say a little bit later, but since you got some momentum started, I will say I didn't realize it had been quite that long. But it's obviously great to have a collaborator who's really talented and energetic, which Natalia certainly is. The other thing that's been really fantastic is the fact that there's increasing overlap, but when we started, there was essentially no overlap in the expertise and knowledge base that we had. It's really easy to collaborate with somebody that thinks like you and knows the same things that you know, but it's actually hugely helpful to work with somebody that has skills that you don't have. And Natalia's lab routinely does things that we didn't even know could be done. We now know they can be done, but we still kind of think it's wizardry. So it's been really fantastic, and I do think it's true that this particular project and the direction that it's going, it's hard to imagine a single lab having the expertise to do that. So it's really been terrific. Okay, so my lab's been interested in the cellular response primarily to xenobiotic-induced DNA damage, but also endogenous DNA damage for a long time in pharmacology. My focus has been changed a little bit to thinking more about anti-cancer and anti-neoplastic chemotherapeutic agents that induce DNA damage. And I'll make a quick reference to that, but I just wanted to get the so what, who cares question out of the way. The long-term goal in our lab, in our lab is to try and understand how cells recognize and respond to this damage, because we now know somewhat surprisingly that the drugs don't really kill the cells. The damage doesn't kill the cells. It's the cellular response and the activation of programmed cell death, and more importantly, the failure of cells to activate that cell death that is responsible for chemotherapeutic failure. So sort of traditional classic pharmacology interested in signal transduction. So we're right now, I'm going to talk about my first love, which is DNA repair, and I'm going to focus on a particular type of lesion that is created by some of these important anti-neoplastic agents, as I'll say a little bit more about in a quick second. Steve pointed out, I need to update my slide. This is the old the old bridge across Washington Avenue, and there's no light rail line there, so I got to get moving on that. So this is a picture that's and it's worth saying this is kind of the biologist's view of structural biochemistry. This is about as complicated of a view that I have of what a DNA protein crossing looks like. I may have another image or so with a little bit more molecular detail. But this review article that Hiroshi Ide and his colleagues published several years ago now, kind of trying to bin these DNA protein crossings into several different categories. And I'll just say the real challenge that we have is it's vexingly complex. I'm sure hundreds, if not thousands, of cellular proteins are able to cross-link, form covalent bonds to DNA, multiple different residues on any one of those proteins, different aspects of the DNA that are attacked covalently by these structures. So what he's trying to do here, and this may work for you, it may not, but he's trying to sort of bin these massive numbers of crosslinks into a manageable subset to think about. So he talks about type 1 through 4. He's the only person that uses this nomenclature, so it's not especially important. But while I was talking, maybe you were looking at this. So most of the xenobiotic-induced DNA protein crossings would be in this type 1 category, and I'll say a little more about that. But I think it's really important to talk about the fact that endogenous molecules, replication and repair proteins, also, with some degree of frequency, we don't know how frequent, can end up being trapped on the DNA. I think we're all familiar with the topoisomerase type 1 and type 2 that can become trapped as part of chemotherapeutic regimens. But DNA repair proteins, cytosine DNA methyltransferase, polymerase beta, other repair and replication proteins can become covalently attached to the DNA. And as I said, they can attach to nitrogenous bases, they can attach to the ribose, they can attach to the phosphodiester backbone with or without single or double strand breaks being present. So again, the idea that I want to make here, just a hugely complex array of lesions. This is from a really terrific review article that Natalia and I didn't see if Arnie and here. Okay, hi guys. So Arnie and Xiaofei are here, and I'll say a little more about them later, but sort of showing some of the structures that are formed, reactive oxygen species, environmental and occupational toxins like formaldehyde, reactive nitrogen species, chemotherapeutic agents, and other molecules that can be industrial contaminants and products are all capable of forming, like I said, this sort of large complex array. We probably know less about DNA protein crosslinks than any other type of DNA damage, unless there's a type of DNA damage that we haven't discovered yet. So over the course of the last, say, 20 years, there's been an increasing effort to learn more about this. And I did just want to say one more quick thing before moving on. I think the only way to really be able to make progress in understanding what these lesions are, how cells recognize them, which repair pathways are involved, and what are the consequences of repair and misrepair of them, is through a combination of techniques. So I'm going to go through some things. I'm going to talk about some published data first, and then I'm going to talk a little bit about our more recent work, some work that my students have been working on, and with help from a number of people in the Tretyakova group. So let me just start with saying the first approach that one can take is to use one of the drugs or agents that one knows induces these agents, treat cells in a variety of ways, and then using mass spectrophotometric approaches, actually look at the consequences, what types of proteins are cross-linked. Nick told me, don't touch this button, which means I probably will touch it. So I'm going to apologize ahead of time. I'm going to try this thing here. So this, again, is another, it's a schema showing what's going on in the experimental approach that, again, the Tretyakova lab looked at to catalog the types of proteins that end up being cross-linked to DNA following treatment with this phosphoramide muster, which is the active metabolite of the important chemotherapeutic agent, cyclophosphamide. So that's the schema there, showing essentially treating the cells, recovering the DNA, and then ultimately doing mass spectrophotometry. It's worth saying, if you still work at the bench or if that's what you do, these are very difficult experiments to do. Techniques have been developed to study protein. Techniques have been developed to study DNA. When DNA and protein become covalently stuck together, very, very challenging. And it's really been it's been wonderful in a way to see the expertise develop, but every new person that comes along in this project, it takes them quite some time to figure out how to do this because none of the off-the-shelf techniques are really adapted for this methodology. So that's sort of my way of saying that as difficult as this work looks, it's actually far more complex and hard to do than these slides kind of do justice. So I think this might be a little bit hard for you to see this image here. Sort of a take-home message. Arnie and his colleagues were able to show over 130 different proteins. So they took cells in culture, a human fibrosarcoma cell line called HT1080, was treated with this phosphoramide mustard, and then methodology was done to recover these proteins. And so again, this is really trying to capture the complexity, and one can spend quite a bit of time looking at this, and for reasons many of us have. So I'm just going to show you through this. You can take a sort of an informatic approach, geo-annotation, to look. So what types of proteins get cross-linked to DNA? A lot of the usual suspects, but quite interestingly, many other proteins that you wouldn't imagine would find themselves in proximity to DNA so they could get cross-linked by these agents do. Again, we don't really know what is the significance of any one of these types of cross-links. The lesion to the DNA per se obviously is important, but one can think about an actual depletion of important nuclear proteins. So even if the cell is not going through division, even if the cell decides to not respond to those DNA lesions per se, and they don't have any proximal toxic effect, depletion of important cellular proteins, so a knock-on effect that may also be important, sequestering and losing important biomolecules. This is an illustration I want to make, again, from the paper, but just wanted to sort of highlight what you can see on the left-hand side of this slide is there's a substantial number of proteins that are cross-linked to chromosomal DNA. So this is a protein gel that one isolates cunningly and with great skill chromosomal DNA that has protein covalently attached to it and able to resolve the DNA off of the protein, run it on a gel. And hopefully what you can see is in the presence of cells that were treated with the phosphoramide mustard, lots of proteins, a broad molecular weight range, significant amount of proteins have become cross-linked to the DNA. But even in the absence of the phosphoramide mustard, there's a significant amount of material. So endogenous processes are associated with cross-linking of proteins to DNA. So this is not just an issue that's important in drug-treated cells, this DNA protein cross-link. Okay, so I think the next slide here, a quote from a paper more than 10 years ago now. Sue Barker published this paper, and the quote, you can read it. This remains a real challenge. The drugs that induce DNA protein cross-links induce a huge number of other types of lesions, and it's really been very, very hard to try and tweeze out, well, how important are DPCs when monoadducts, cross-links, double-strand breaks, reactive oxygen species are being generated in these same cells at the same time. So quite difficult to do that. So really quickly, because I don't want to spend a huge amount of time on data that's really a couple of years old now, but we did take an approach to ask the question, could we design an agent that was uniquely capable of inducing one type of DNA damage alone, and that is DNA protein cross-link. So the strategy that we used came out of a collaboration. I think this is when Natalia and I really first started working together. There was an interest in asking the question whether this DNA repair protein, 06-alkyl-guanine DNA alkyl transferase, it's called AGT. Some people call it methyl-guanine methyltransferase. So it's a DNA repair protein. It's worth saying it's not a repair enzyme. The reaction scheme is shown there. This molecule scans along DNA, finds methylated or other alkylation damage on the 06 position of guanine, and is able to catalyze is able to basically transfer the lesion, the methyl group, off of the DNA onto the protein. You look at this reaction for a while as a biologist, and it's like, ooh, that's kind of cool that you can do that. But the fate of that protein is it then undergoes proteasomal degradation. So its turnover number is 1. It's not an enzyme. It's really difficult to imagine the evolutionary pressure that would create a protein that had one job in life, which was to kill itself by removing a methyl group off of one guanine residue. But this is an evolutionarily conserved protein, been around for a long time. Make a long story short, Natalia and her colleagues, and eventually I, became interested in whether this protein could also become covalently cross-linked to DNA if in its effort to remove a methyl group, say, from an 06 position, it were able to become covalently attached to a bis electrophile that had attacked this N7 position. Long story short, that could happen. We were able to show, my colleagues were able to show that, what came first, the chicken or the egg? Did this protein, did the bis electrophile attack the DNA first, and then the AGT got modified? That could happen. Could it go the other way about? Yes, it could. We discovered that you could take a molecule, diaproxybutane, incubated with the AGT molecule, and it would form one of the epoxide residues would attack that reactive sulfhydryl group on the AGT. And interestingly, and in a way that this biologist doesn't really understand, that molecule was stable enough that it would retain that reactive epoxide residue for a significant period of time, long enough, in fact, for us to prosecute the experiment that I'm now going to quickly run through here, and then you'll see the logic of why I've been going into this. So we wanted to find out if we could, in fact, take a reactive, what I'm calling AGT monoepoxide, so that I think the chemistry is sort of shown here. Diapoxybutane reacted with the AGT molecule. This is that reactive sulfhydryl group, this reactive monoepoxide. This is a stable molecule. We were able to purify that away from unreacted diapoxybutane, electroporate that molecule, the protein AGT molecule, electroporate that into cells, and then ask the question, what happens? Now, it's true that this molecule wouldn't just attack DNA. We knew that it could because we were able to incubate this reactive monoepoxide molecule with synthetic DNA substrates in vitro and show that the cross-linking happened. So we knew that the chemistry could happen. Did it happen in cells, and what was the consequence? And just as a caveat, I'll say, could this AGT monoepoxide react with other cellular macromolecules or small molecules? The answer is yes, indeed, it could. I'm going to show you some data real quickly and make the argument that these data suggest that, in fact, DNA-protein crosslinks are cytotoxic and mutagenic in human and hamster cells. So without going into any detail, I'll suffice it to say that Aaron Michelson-Ritchie, who was a master's student in Natalia's lab, terrific person. When our labs were closer together, you almost couldn't tell where Natalia's lab ended and mine began. Now we know it's like the transitway is where they end. But still, her folks come over to my lab quite a bit, and we're over in this building quite a bit as well. And so Aaron was actually spending a lot of time in my lab doing these experiments. I did a few of these experiments. And what you can see is in this HT1080 human fibrosarcoma cell line, as well as in a hamster, Chinese hamster lung fibroblast clone, electroporation of this molecule, the reactive monoepoxide molecule, into the cell induced cell death in about 20% of the cells. And suffice it to say, we were able to show that the protein itself had no biological activity, the molecule that we used, and that there was no free monoepoxide contributing to this toxicity. So this is toxicity above and beyond any toxicity associated with electroporation. So in a hamster background and a human background, we were able to show that a molecule that would be capable of forming DNA-protein crossings was indeed cytotoxic. We were able to take advantage of the fact that there's a version of this AGT protein that has a single nucleotide substitution in it, a lysine to leucine substitution at 125 amino acid position. This particular molecule is incapable of accumulating in the nucleus. So we were able to ask a simple question. If we form this reactive monoepoxide molecule on a version of the AGT protein that couldn't get in and stay in the nucleus, would it still be toxic or not? And the question and the answer is that this molecule is, in fact, essentially non-toxic. There's a little bit of toxicity here, but this is not significantly higher than the background level, and it is significantly lower than the toxicity here. So while these data don't directly show that DNA-protein crossings are responsible for the toxicity, it does say the target for this reactive monoepoxide protein is in the nucleus. And furthermore, to show me, who's no longer here, using the Tretyakova Lab's wizardry, was able to use mass spectrometry, in fact, to show that in this essentially wild-type version of this protein, this conjugate that is the actual molecular signature of that DNA-protein crosslink, this signature molecule is only formed in cells in which this reactive monoepoxide can get into the nucleus, which is what you would expect to see. And again, showing both cytotoxicity and the formation of this conjugate. So while I think one may be able to think of other potential explanations, I think the overwhelmingly favored interpretation of these data is, in fact, that these DNA-protein crossings are cytotoxic. And then really quickly, just to finish this up, data showing that mutagenesis at a locus in the cell, the HPRT locus, when it becomes inactivated, renders cells resistant to 6-thioguanine and able to show, as one would predict, if these lesions are in fact mutagenic, in addition to being cytotoxic, you see a statistically significant increase in levels of 6-thioguanine resistance in these drug-treated cells. So, long story short, I think these are our data to show that the so what, who cares question. Indeed, DNA-protein crosslinks are cytotoxic and mutagenic. We haven't really done very much more with this. If I had time, I would go into a little more detail, show you some images. I will just say real quickly, we're trying to develop other types of reactive monoepoxide molecules. We'd love to be able to target these molecules to specific loci in the chromosome so we could actually look at the molecular signature of what these DNA-protein crossings are like. We're left really with global averages, making statistical arguments. We'd like to be able to drill down a little bit closer to do some real molecular cellular biology, and that remains an aspiration at this point in time. But so, like I said, I'd talk about three general approaches taken to study DNA-protein crossings. The first was cataloging the types of protein DNA crossings that are formed when a variety of different reactive molecules are used. And in the interest of time, I didn't really want to get into this, but I would say that the Tretyakova group, and we've helped them a little bit, not very much, we've helped them a little bit, looking at other types of reactive molecules. And it's interesting, the types of proteins that become cross-linked to DNA if you use diapoxybutane or cisplatin or the phosphoramide mustard or other molecules. There's some overlap. There are some proteins in common. And interestingly, there are some proteins that are only cross-linked by one agent and not another. And I'm sure that that's interesting and important. Again, exactly how we're going to figure that out remains sort of a challenge. But I think it has some potential significance trying to understand to what extent these therapeutically important molecules are DPCs contributing to off-target effects, to adverse effects, organ-specific toxicity. We do know DNA-protein crosslinks form spontaneously in the bodies of living organisms. I haven't done any of those work, but we do know that happens, and there's every reason to believe that some adverse biology is being driven, or pathology is being driven by these spontaneous DNA-protein crosslinks. So the last thing I'm going to talk about, and I'll spend the most time talking about it, is this third strategy that we've been using to try and study DNA-protein crosslinks, and particularly DNA-protein crosslink repair. So some traditional, sense methodologies. The logic is quite simple. Prepare a defined DNA lesion on a plasmid molecule and introduce that plasmid molecule into the cell. There's some tremendous strengths associated with doing that. If you have the skill and the patience and the ability to create large numbers of high-quality repair substrates, you can put them into a variety of different cellular backgrounds and track fates. We're very excited about being able to do this with extracts. I'm sorry to say I don't have any data for you today, but the possibility not just of putting these lesions into cells and following their fates, but being able to trap reaction intermediates if we can either pull substrates back out of cells or use extracts to study that. So the strengths of this approach are overwhelming, and it's worth saying most of the detailed molecular biology that we understand about DNA repair has come from these types of strategies. I might frequently get asked, and I'm not at all disappointed or upset by critical commentary, how important or relevant is DNA repair on a plasmid when what you're really interested in is DNA repair on a chromosome? I think it's a really fair question, and if you kind of visualize it and think about those different molecules, it would be logical or reasonable to suggest, indeed, there might not be a lot of overlap between the biology, the way that a cell approaches a 5,000 or 8,000 base pair circular plasmid molecule versus human chromosome number 1, right, which is millions of base pairs in length. The reality is, I don't think one can point to a single example of a type of reaction that happens differently on these extra chromosomal plasmid molecules compared to what happens on the nucleus. In other words, when models have been proposed based on extensive testing in plasmids, subsequent efforts have shown the same types of reactions occur at the chromosomal level. So I'm not trying to be defensive about this, and I'd be very happy to talk about this later on, but this is a strategy that we and others have used, and it's the approach that I want to talk about right now. So I'm going to go through some images and kind of bear with me a little bit, some schematic diagrams that may or may not work especially well for you. The first lesion I'm going to talk about is and when I say we, this is now work that Lisa Chesner, who's sitting right in front of me, has done. Synthetic oligonucleotide that contains an oxoguanine residue within it, annealed to a single-stranded M13 molecule. So if you remember your university molecular biology, you may have learned bacteriophage, a small mobile genetic element that infects E. coli called M13. It's about 8,000 bases in length. It can exist in a single-stranded or a double-stranded form. We either purchase or purify, depending on our finances and our enthusiasm or energy, I guess. Purchase or purify single-stranded M13 circular molecule, anneal this oligonucleotide to it in the presence of a polymerase and a ligase and nucleoside triphosphates and a few other cofactor requirements. This polymerase will extend. So this is a square piece of DNA, apparently. So to see primer extension, and it'll go all the way around the molecule, forming a full-length duplex molecule. DNA ligase will seal that nick. And so what you can kind of see here, hopefully this is an image of a molecular ladder. So it's an agarose gel stained with ethidium bromide, just to show the single-stranded circular molecule gratifyingly runs faster than the double-stranded molecule, which you would sort of expect. Interestingly, if you, again, remember your physical biochemistry, you know that this type of molecule here, the way the coach drew the play on the board, would be 100% present in what we call this covalently closed circular form. So a duplex molecule with no nicks in it, supercoiled. This molecule can also exist in what they call an open circular form. If there's a single nick in that DNA, the DNA will unwind, and if you run this gel in the presence of ethidium bromide, it'll run it. So these are three basically topoisomers. This is a double-stranded linear version. This is a covalently closed circular version. And this is the open circle or relaxed circular form of the DNA molecule. If the reaction were driven 100% to completion in the way that we think it should, and the way you draw it, 100% of the DNA would co-migrate with this supercoiled double-stranded M13 molecule. That doesn't always happen. It's actually quite difficult to drive these reactions exactly the way that you want to. But we're able to generate enough of these molecules to actually purify this covalently closed circular molecule. So now we have a duplex molecule with a single oxoguanine residue present exactly where we want it to be. We did not invent this chemistry by any means. Others have shown oxoguanine glycosylase. This is the normal reaction mechanism, kind of interesting part of the base excision repair pathway. I've only showed the first step in it, the first recognition, generating basically an A basic site. The molecule, the DNA repair protein, importantly, forms what's supposed to be a transient cross-link to the DNA substrate, not at all an uncommon event. This then spontaneously hydrolyzes back off, releasing the DNA and the free enzyme able to catalyze further reactions. You can trap this molecule by adding a strong reducing agent, sodium borohydride or cyanoborohydride we use. You can form a borohydride complex. This is a covalently cross-linked DNA protein molecule which is inherently stable. It won't move out of this form. So long story short, Lisa is able to essentially generate a well-defined DNA protein cross-linked molecule in which the human oxoguanine glycosylase molecule has been cross-linked onto this duplex DNA molecule. So two different techniques, just to if you don't believe what I'm telling you, there's some evidence to support this. Interestingly, and this was an observation that was made, I think, as far back as the late 70s or early 80s. Sodium SDS, as we all know, solubilizes proteins. Potassium SDS is actually insoluble, so one can precipitate DNA that has protein attached to it if you first add sodium SDS and then drive the sodium away, but with an excess of potassium. So we call this KSDS or potassium SDS precipitation. So this molecule, Lisa made this molecule. She cut it with a restriction enzyme that generates two fragments, a smaller fragment that has the protein attached to it and a larger fragment that does not. You then add potassium SDS, and if most of the DNA actually does have the protein cross-linked to it, which is an essential requirement for us to prosecute these experiments, then the smaller of these, or the larger of these two bands should quote-unquote disappear. It doesn't disappear. It becomes insoluble. It's actually stuck up in the well of the gel. You can't see it. So what you can see here is a small shadow of unreacted DNA, meaning that this covalent cross-link is not 100% efficient, but the efficiency of this reaction is quite high. One can also just do an electrophoretic mobility shift experiment, which is a little bit easier to understand, but is technically a little bit harder to do. Again, using restriction endonucleases to generate fragments of DNA. A small portion of this duplex molecule with the protein cross-linked to it in the presence of the protein will run slower on the gel, appear higher up on the gel. And again, you can see this one actually looks nicer in a way. It looks like it's 100% cross-linked. That I think there's just some technical limitations with respect to background. But we're getting well over 95% of the substrate being converted to product. And Natalia has a lot of fun calling me a dirty biochemist. But in our lab, 95% is, exceeding all expectations. So we're quite happy that we in biology are much more comfortable living with background, dealing with background, and so we have appropriate controls to get around that. But it's worth saying, again, the huge challenge with these experiments is being able to generate sufficient quantities of what I call high-quality substrate. We can deal with a little bit of background, but we have to have and know what we've got. And so it's taken us really years and years and years to develop this methodology. It's quite challenging. So this is a kind of upsetting or startling image to show. And I won't dwell on it for very long. But so having produced this substrate, we want to ask a really simple question now. If we put this repair substrate into normal or wild-type cells and measure a repair efficiency, would that efficiency be different if we went into a background of cells that had a deficiency in a known and characterized DNA repair pathway? And the one that we've chosen to focus on is nucleotide excision repair. I've got a really complex slide I'll show in a second. Suffice it to say, nature has endowed us with this curse or gift of human individuals who have inherited through an autosomal recessive genetic pathway a defect in nucleotide excision repair, and what you can hopefully see here, and I'll get off of this in just a second, even exposure to very modest levels of sunlight causes massive skin lesions. These individuals are exceptionally prone to skin cancer, and interestingly, other types of cancer as well, including cancers of internal organs, which really hard as a biologist to understand how your internal organs are getting sun damage. So there's something more than just repairing photoproduct damage associated with defects in this pathway. So this is the so-called NER pathway, and I'm as prone as every expert to devolve into acronyms and abbreviations. So please forgive me. NER, nucleotide excision repair. So we're basically testing a hypothesis. Is the NER, is the nucleotide excision repair machinery involved in the repair of DNA-protein crosslinks? And we would deduce that or support the hypothesis by comparing the frequency with which repair events occur in a wild-type background versus a nucleotide excision repair deficient background. So that's kind of the logic. But I wanted to run through just real quickly. So this is the experimental design. Quite simple. Again, we have sufficient amounts of this substrate that we can introduce them using, in these experiments right now, lipofectamine. I'm also actually quite excited Lisa has perfected the ability to use electroporation, which is a physical technique as opposed to this sort of quasi-chemical and biological technique. Much faster to get the DNA into the cells. The route of transfer is almost instantaneous, and it doesn't involve going through endosomal pathways. So we're quite excited about the electroporation approach. It's just a very inefficient way to get DNA into cells. And Lisa looks at me like I'm a vampire when I ask her to do electroporation experiments because you have to start with like 20 times as much DNA. But that's I hope the direction that we're going, Lisa. Anyway, so the results I'm going to show right now have been done with this liposuction strategy. So going into the V79, just the title of these cells, these are Chinese hamster lung fibroblast cells, the wild type cell, and an isogenic derivative from that, which has a defect in the XPD gene. So we introduce the substrate, pull the substrate back out again. Now you got to kind of bear with me as I go through this slide, which once you get the hang of this, it's totally simple to understand, but it's a little bit complex at first. So how are we going to we're going to put the substrate into the cell, wait a period of time, pull it back out again, and have some way of quantitatively measuring whether repair has occurred or not. And the strategy that we're using is what I'm calling a asymmetric primer extension quantitative PCR strategy. So the first step is to denature the DNA and then anneal a primer to it. So this is primer B, calling it primer B right here. This again is my physical biochemist's description of a DNA-protein crosslink. What we know is that this DNA-protein crosslink will block primer extension. So you can anneal the oligonucleotide, you add the polymerase is unable to extend that primer through this lesion. So in the presence of this DNA-protein crosslink, you basically block the transit of the molecule. You don't generate any full-length product. So what we do is I don't do this, Lisa does, 8 sequential rounds of primer extension. So you extend, you anneal, extend, denature, anneal, extend. So it's kind of like PCR, but you only have one primer in there. There's primer B, no primer A. This is the ultimate PCR primer that we're going to use, but there's no primer A in these primer extensions. You're extending primer B. 8 rounds of this. So if you have repair, you will generate 8 additional strands of this, Crick. I call them Watson and Crick. So you make 8 strands of Crick. There's already the one when you denature it. I'm not showing that. You can make 8 additional ones if you've had 100% repair or if you start with an undamaged substrate. The subsequent step, of course, is then to take the product of those primer extensions and then do quantitative real-time PCR. Many of you in the room have done this. It's a technique that allows you in real time to monitor the product formation from a polymerase chain reaction. We now have primer A and primer B. And this important concept and the most important thing you want to think about here is this so-called CT value. This is the threshold. How many cycles, the cycle threshold, how many cycles do you go through while you're in this sort of linear phase of the polymerase chain reaction? When you get to that inflection point or where you arbitrarily set that, the more substrate you start with, the lower your CT value is, which is a little vexing. So if you have a lot of substrate, it doesn't take very many cycles to reach a threshold. If you have not very much substrate, it takes a long time. So we're then able to basically calculate the amount of repair that we see by generating this, what we call this delta CT value. So we can essentially convert the data from comparing what happens when you've done 8 rounds of asymmetric primer extension versus when you didn't. So the same material is split in two. One part goes through the primer extension, one part does not. The relevant or respective CT values are calculated. You subtract this, you can then calculate the delta CT, and from that you can estimate the percentage of DNA repair that's gone on. So again, it's a little bit complex, a little clunky to talk about, but it's essentially a methodology that allows us to quantitate DNA repair based on this primer extension. So we've incorporated a positive control assay, which I think is kind of clever in a way. If you take the 8-oxoguanine residue, so the non-cross-link material, I think I told you we start with the 8-oxoguanine residue on there, and we use borohydride to trap oxoguanine on there. If you don't add borohydride, the oxoguanine glycosylase simply removes the 8-oxoguanine residue and generates an A-basic site. It turns out that A-basic site will also block primer extension. So we can, in effect, do something that others have done in a much simpler way decades ago and use a very complex assay to measure base excision repair. So as a kind of a positive control, we measured base excision repair in the wild type and nucleotide excision repair deficient clones, and we should have gotten the same result because there's no defect in base excision repair in nucleotide excision repair deficient clones. So kind of a positive control that we did, and that, again, Lisa did. And gratifyingly, what you'll see is no difference in, essentially no statistically significant difference in the repair kinetics of this 8-oxoguanine residue in the VH1s and the V79 or wild-type clones. I mean, indeed, it actually looks like the VH1s are a little bit better at that. I don't think that that's true. This is the average data from n equals 3, so you can see the efficiency of repair is quite high, and the time course is quite fast. This is an hour and a half post-liposuction. So maybe an hour and a half sounds like a long time, but it takes quite some period of time for the lesion to actually get into the nucleus. So this is reflecting something which has been known forever, which is that base excision repair activity is very robust and efficient and is not deficient in clones that have this nucleotide excision repair defect. So another sort of, if you call that a kind of a positive control, we also want to do a different type of control. It had been discovered some time ago, again, about 15 years ago, that one could generate a synthetic lesion in which a cholesterol molecule had been cross-linked onto a nucleotide base, and there are phosphoramidites that are available. So we were actually, I think we purchased this molecule. We didn't make this molecule. So an oligonucleotide that has this cholesterol moiety attached to one of the nucleotide residues, interestingly, is a substrate for nucleotide excision repair. I don't think anybody believes that cholesterol actually gets cross-linked to our DNA, although that maybe that's true. The nucleotide excision repair pathway is specialized at repairing DNA photoproducts, but it's also capable of repairing a large number of so-called bulky adducts, and this would be one of the bulky adducts that can be repaired. So this is an experiment, a figure from this paper, which I've cited here, showing essentially the cleavage and releasing of this portion of the substrate that actually has the cholesterol attached to it here. So the nucleotide excision repair pathway, I didn't drill down in this because it's not especially important, but the way this reaction works is nicks are essentially induced on either side of the lesion. The helicases remove a portion of the DNA, the strand with the DNA on it, and it's somewhere between 26 and 34 or so bases in length. Interestingly, it's not precise. It's not like it's exactly this many nucleotides downstream and upstream of the lesion, but it's in that range. So this is just basically showing that this cholesterol substrate is actually acted upon by the nucleotide excision repair machinery. So if that were true and everything else that I said was right and we actually understood what we were doing, we should be able to use the same sorts of approaches that Lisa used to make a DNA protein crossing to make a cholesterol DNA adduct on this duplex plasmid, introduce it into the V79 and the mutant NER deficient cells, and see reduced kinetics of repair. And indeed, with that setup, you could have predicted that that's what happened. So this is showing repair of a DNA cholesterol lesion in wild type in blue, and these VH1 XPD nucleotide excision repair deficient clones. And there does look like some small level of repair going on, but I think that, and this again is an N equals 3, I don't believe that the results here are statistically significant. So there's a small, what looks like a level of repair, which is probably not really significant. So finally set up to actually do the experiment that I've been sort of spending 15 minutes setting up here to look at the repair of a DNA protein crosslink in wild type and VH1 nucleotide excision repair deficient clones. And this is sort of, this was a really good day in the lab. This is an experiment that had been maybe 5 years in coming, incremental progress to set this up, and just really thrilled to be standing here in front of you talking about these data. So a longer time course here. So I think this is 3 and 8 hours post liposuction, and what you can see is linear kinetics of repair of this crosslink in the wild type cells, and again, a level of repair that will be consistent with background or no repair going on in these nucleotide excision repair deficient clones. So we're pretty close to the end of the data. I think I just have, well, yeah, that pretty much is the end of the data. I have one more control experiment that I want to talk about. So these experiments, again, have been done in a particular clone that has a defect in one of the nucleotide excision repair genes in a Chinese hamster lung fibroblast background. Naturally, we'd like to repeat that in human cells, and this is just the first control experiment that Lisa's done, showing that a human XPD deficient clone and the corrected clones show the expected phenotype with respect to repair this cholesterol lesion. What I wish I had right now was the data from DNA protein crosslink in those human cells. I do not have that. That's an experiment that's ongoing right now. Last 2 or 3 slides, I'm just going to hopefully leave some time for questions here. Wanted to show some of the other strategies that we're using to try and generate these clones. And by I say we now, really this is the expertise of the Tretyakova lab. I see Suresh, another one of our colleagues, has come in. So I'll say a little bit more about these 3 gentlemen in just a moment. But so trying to use cunning chemistry to generate different types of DNA lesions, DNA protein crossing. So one of you might have said to me, and still might, well, you're studying a very particular type of DNA protein crossing that's probably not representative of most of the types of lesions that are formed, certainly in cancer cells that are being exposed to anti-cancer agents. And I would say that's exactly right. We've started with the low-hanging fruit. That's my favorite place to start. We're trying to use other types of chemistry that would allow us to put different types of proteins, have a little bit more flexibility. This is simply showing one of the strategies, this reductive emanation strategy that we're hoping to pursue. We've had some success, and again, these experiments are much more difficult to prosecute than one would anticipate. I think we've been disappointed. A huge amount of ingenuity, almost none of it mine, but a huge amount of ingenuity has been just destroyed through beautiful ideas, destroyed by ugly realities and difficulties that were unanticipated. So again, trying to develop some methodologies, some synthetic strategies, using recombinant proteins, using oligopeptides, a variety of different strategies. Just really quickly, we're very excited about another particular approach here, different type of chemistry used to generate lesions on a different type of nucleotide base. And then just really quickly here, because I do want to leave some time for questions. So where are we right now? First goal, look at DNA protein cross-link repair of this oxoguanine glycosylase lesion in human XPD deficient clones. If I had more time and if I thought it were really helpful, I'd go over the fact that this nucleotide excision repair pathway has two main arms. One is a transcription coupled, and the other is what they call a global genomic. The substrate that we're using neither replicates nor undergoes transcription. So we're looking at a lesion or a piece of DNA that you might consider to be fairly boring, although I would hasten to say in most of our cells, most of our genome are transcriptionally silent, and those cells are not dividing. So that actually may be more representative of most of the loci that undergo DNA protein crossings in our bodies. But we're very interested in asking the question, now that we have data that suggests the global genome NER pathway is able to repair these lesions, how does it do that? So we'd like to drill down, ask the question about global genomic versus transcription coupled nucleotide excision repair. We'd really like to look at the effect of transcription, which again would tie back with this global transcription coupled nucleotide excision repair. We'd like to look at the effect of replication. The kind of lore in the field, if you will, is that DNA protein crossings are only repaired in the context of replication. We know that's not true now. Granted, we're using a different type of substrate, but very interested in trying to ask the question, is the rate of repair influenced by the presence or absence of the replication machinery? I didn't write it down here. We'd like to look at, are these pathways error-prone? There's some strategies that we could use to look at that. We'd like to try and look at other DNA repair pathways. We're particularly interested in the Fanconi anemia-dependent recombinational repair pathway, which I think is everybody's favorite guess of how these lesions are repaired. We're very interested in whether cellular proteases, particularly Spartan, play a role in modifying or converting DNA protein crossings into peptide crossings, and can we trap intermediates? And the final thing I'll end with, my first love in getting involved with DNA repair was in vitro repair in extracts and reconstitution studies, and that is kind of the long-term, extremely ambitious goal here, is to be able to trap reaction intermediates and really learn more about the molecular mechanisms. Because, again, just to finish where I started, the long-term goal here is not to understand the repair pathways that are involved, but what are the consequences of misrepair? How are the ways in which different types of therapeutically important lesions are recognized, and the failure, what is the role of failure to repair? How does that correlate with clinical failures in people that are undergoing cancer chemotherapy? I'll stop right now, and I will be happy to take questions. Thank you.
So in an early slide, I noticed that there were mitochondrial proteins listed, and so it made me think about mitochondrial DNA protein adducts. And as I understand it, mitochondria don't have nucleotide excision repair, so I wonder if that's particularly toxic to cells, and if you've thought about trying a mitochondrial targeted protein that's been modified in the way you did ATG to see if that's actually a toxic. Yeah.
I love that question. I really appreciate it. One thing I would just say, those geo categories are wrong a lot. So when they say a mitochondrial protein or a cytosolic protein, that means it's known to be in the mitochondria. Doesn't mean that it was in the mitochondria when this thing happened. But man, I just love thinking about mitochondria. I spent way too much time thinking about mitochondria. I'm super interested in mitochondrial DNA repair. It's just the way you really want to do that experiment is put some of our lesions into the mitochondria. And, that's really, really challenging. I would love to get back to that, and I think you're exactly right. Well, we have some data about 15 years ago where our lab was really focused on mitochondrial DNA repair. I think mitochondrial DNA damage may be playing a much bigger role in therapeutic outcomes than we know, and yeah, I think that could very well be. I would love to look at that. Maybe if we can figure out how to do extracts, we can make mitochondrial protein extracts and do that. But that's going to be an aspiration for a while.
Other questions?
Let me just, because I'm horrified, I forgot to do this. So Lisa's right here. I haven't mentioned people along the way, but Lisa's right here. Petra couldn't be here. Arnie, right in the, well, it's the second row, but that counts as the front row if you're a grad student. Xiaofei is next to him. Suresh is in the back. These three gentlemen, I just have to say it's been a real pleasure for me to kind of be in the room and watch graduate students teach each other. I think it's probably like in some ways the most rewarding part of this collaboration is the bringing people together and learning, and Lisa helping these gentlemen understand some of the nitty-gritty details, some of the biology. A lot of learning going the other way. Us learning the chemistry, not just Lisa, me learning the chemistry. These guys are very, very talented people. There's other people from Natalia's group that are in the room, and we have worked together for many years, but they have not been involved in this particular project. But just to say, Arnie and Xiaofei and Suresh, it's really good to have you here, and publicly thanking you for all your help.
Thank you.